Hello, welcome to my presentation, a representation method for cellular lines based on support vector machine and text mining. I'm Iván Carrera. I'm a professor of the Departamento de Informática y Ciencias de la Computación of Escuela Politécnica Nacional in Quito, Ecuador. And my co-authors are Inés Dutra from Departamento de Ciencia de Computadores of Universidade do Porto in Portugal, and Eduardo Tejera from Grupo de Quimio Bioinformática of Universidad de las Américas in Quito, Ecuador. My presentation is divided into three sections: Introduction, Methodology, and Results. Research into cancer and other life-threatening conditions relies on culture of cell lines as effective models for the cell type being studied. Computational methods for predicting of sensitivity of cell lines to chemical compounds is important to optimize in vitro assays in the drug discovery processes. There are two major groups of approaches on cell line description for drug discovery. First, based on sensitivity profile against chemical compounds, and second, based on gene expression. Approaches based on QSAR modeling aim at establishing a model relating chemical structures with their effects on different cell lines. This means that for every cell line, we have to have a sensitivity profile relating chemical structure and chemical compounds. Approaches based on gene expression compare the genetic expression profile triggered by a query compound with a known reference expression pattern generated with a particular cell line compound pair. These methods require the gene expression profile triggered by the compounds under investigation, which is frequently unknown. Representation of cell lines could be based on text processing, given that the scientific literature is considered as the primary repository of biomedical knowledge. There's a need for a representation method for cellular lines that can be computed in silico and can be general enough as to be utilized with a large number of cell lines. We present a method for representing cellular lines based on support vector classification and text processing of scientific literature. We form a corpus of scientific paper abstracts related to the cellular lines retrieved from PubMed. Corpus was transformed using stemming, TF-IDF vectorization, and feature selection. A support vector machine document classification algorithm was trained for obtaining the hyperplanes as decision boundaries between sets of documents, or in this case, between descriptions of cellular lines. Every class representing every cellular line is characterized as a support vector descriptor. Our goal is to understand how to obtain an unambiguous characterization for cellular lines for published scientific literature. A cell line is a population of cells from a multicellular organism that, given appropriate surroundings, can live, multiply, and even express differentiated properties in a tissue culture dish, used to perform in vitro experiments. Important role in examination of stepwise alterations in the structure, biology, and genetic makeup of the cell under controlled environments. A good characterization and representation of cell lines is important to allow for automatic discovery of cell line interactions, as well as their relations with other entities, and for understanding the role of cell lines in diseases. Various efforts have been made to properly document, characterize, and represent cell lines, such as the American Type Culture Collection, the Korean Cell Repositories, the Japanese Collection of Research Resources Bank, or the Chinese National Infrastructure of Cell Line Resource. We propose a method to automatically characterize cell lines from the scientific literature. The method is based on the support vector domain description introduced by Tax and Drain. Support vector domain description is based on support vector classifiers and obtains a spherical boundary around the dataset in order to describe it and to identify its outliers. Support vector domain description is not focused on distinguishing between classes of objects, but on providing a description to a set of objects. For data domain description, not the optimal separating hyperplane has to be found, but the sphere with minimal volume containing all objects. Whereas Support vector machine find lines or hyperplanes either in the original data space or more usually in kernel space to separate classes. Support vector domain description tries to find a circle or a hypersphere that encloses a class. We pass now to the methodology. This is the pipeline of our work. First, we identify cell lines from Cellosaurus, then we retrieve abstracts from PubMed, then we form the corpus, tagging data, the cell name to the abstract. Then we transfer the data, first with TF-IDF vectorization and second with feature selection. Then we perform a document classification algorithm. And finally, we have the cell line description using support vector data description. Identifying cell lines. The Cellosaurus is a knowledge resource on cell lines. For each cell line, it provides description information, cross-references, and literature citations. As an example, in the figure you can see the Cellosaurus cell line SF295, which has reported 28 PubMed references. For retrieving abstracts, we use PubMed. PubMed is a free resource developed and maintained by the National Institute of Health. It comprises over 30 million citations for biomedical literature. We retrieve abstracts from querying PubMed Search API. In the figure, it's an example of retrieved abstract for cell line SF295. Now we form the corpus. Abstracts are tagged with the cell line identifier for Cellosaurus as shown in the figure. This is the class of the document in the document classification algorithms. The final datasets contains over 83,000 abstracts, and we retrieve information for over 1,200 cellular lines. Then we transform the data. Abstracts processing includes stemming the terms and transforming into TF-IDF vectors. We tested the difference of the classification method when stemming and when not stemming the terms of the abstracts. Dimensions of the TF-IDF vector representations are reduced using feature selection. In the table, we enumerate the number of the dimensions we tested the document classification method. We have chosen 4 thresholds for the dimensions of support vectors: half of a median, the median, median and a half, and 2 times the median. Now we see the results. From a grid search cross-validation, we compare the microaverage precision for different classifier configurations. In the figures, we can see for non-stemmed corpus and the stemmed corpus 4 configurations, changing 2 parameters, the parameter C of the SVM and the parameter of the kernel. The best performance is when the C equals 10 and the kernel is linear. For the train and test set, we test the microaverage precision for the non-stemmed corpus and the stemmed corpus. We can see that for the non-stemmed corpus, we have a better performance. When having cellular lines with 100 and more documents, we have the best performance. For example, for the train dataset, we have a performance for microaverage precision of almost 90%, and for the test dataset for 70%. We have identified 2 constraints. First, to limit the number of cell lines. Filtering cell lines that report more documents, we improve classification performance. Every cell line contains more information and there are less classes for the classification method. However, this makes the characterization method less general. Also, we have identified some unspecific names for the cell lines. For example, cell line H-4 has synonym H4. When querying PubMed search, this will return results that will not correspond with the cellular line, as a string H4 can mean so many other things. Conclusions. We presented a representation method for cellular lines based on support vector machines and text mining. We were able to characterize and represent cell lines by text mining the scientific literature. Classification methods for documents have shown a micro-average precision of above 70% in test datasets. To the best of our knowledge, this is the first attempt for characterizing cellular lines from processing the scientific literature. In the figure, we have an example of support vector representation for cell lines. The representation of cell lines as vectors gives us a tool for exploring the relations among cell lines and other biological entities. The constraint of this work is that if we aim for a very accurate depiction of cell lines as well as precise differentiation between cell lines, we need to select only cellular lines that report a large corpus of documents. Thank you.